In today's lesson, we're going to learn about bacterial transformation and what that means. So typically when we think about inheriting genetic information, we think about genes being passed from parents to offspring. But today we're going to talk about several different ways that bacteria share their genetic material. So genetic information can be transferred vertically or horizontally in bacteria. So first I'll explain what's meant by vertical gene transfer in bacteria. So bacteria reproduce by binary fission, which is asexual reproduction. And what happens is the bacterial chromosome is copied and the cell divides into two. And these two daughter cells are identical copies of the parent. So this mechanism of gene transfer is called vertical because the genes pass directly down from parent to offspring. But if bacteria reproduce by making exact copies, then how do they have so much genetic diversity? Well, horizontal gene transfer can explain that. So horizontal gene transfer is when bacteria transfer DNA from one bacterial cell to another. So in this case, it's going from donor to recipient. And that allows bacteria to quickly acquire new genes for adaptation and survival. So for example, if a normal bacterial cell that's susceptible to an antibiotic receives a piece of DNA that codes for antibiotic resistance, then it will become antibiotic resistant too. So the problem that we see in hospitals and in the medical field, the problem of multi-drug resistant pathogens, it's an example of how prevalent that horizontal gene transfer can be. In this case, spreading a gene from a donor to a recipient to make that recipient also resistant to the antibiotics. There are three types of horizontal gene transfer, which we're going to go over today. The three types are called conjugation, transduction, and transformation. So these are mechanisms of gene transfer that are called horizontal because the genes pass across from donor to recipient, not down from parent to offspring. So the first type of horizontal gene transfer that I mentioned is conjugation. During conjugation, two bacterial cells make physical contact, and the donor cell directly transfers the DNA to the recipient cell. So the donor cell produces a conjugation pilus that connects it to the recipient cell, and the DNA then enters the recipient cell right through that conjugation pilus. Transduction is the second type of horizontal gene transfer, and it's when a virus carries a DNA fragment from one cell to another. So a bacteriophage is a specific type of virus that attacks only bacteria. And the third type of horizontal gene transfer is called transformation. And transformation involves the uptake of exogenous, which means outside or free DNA, from the surrounding environment and takes it into a bacterial cell. So once the DNA is taken into that recipient cell, the genetic information from that DNA is then expressed in the recipient cell. So an example of this is that pathogenic bacteria or bacteria that cause disease can actually transfer their virulence genes from one bacteria to another. And that was something that was first discovered in 1928 by a scientist named Frederick Griffith. So this is a figure of Griffith's experiment. And what he did was he was working with two different strains of Streptococcus pneumonia. An S strain, which is a virulent or deadly strain of it, and an R strain, which is a non-virulent and harmless strain. So when he injected mice with the virulent S strain, the mice would die. when he injected mice with the R strain, the non-virulent strain, the mice would live. So then what he did was he took that S strain and heated it to kill the bacteria. And then he injected the mice with the dead strain of the dead S strain. And since it was killed, the bacteria were killed, when injected into the mice would live. So next, he mixed the two. He mixed the living R strain, so the living non-virulent strain, the harmless one, and mixed it with some of the dead S strain that he had killed with the heat to kill the bacteria. And when he mixed those two together and injected into mice, the mice would die. So that showed that there was a transforming principle that would transfer from that dead S strain, the virulence property of that S strain, transferred to the R strain, making that R strain deadly, which killed the mice. So that experiment showed that transformation is possible, and the R strain was taking up the genetic information from its environment, from the dead S strain, and taking it into its own cells and expressing it and becoming a pathogenic bacteria. So that experiment shows that transformation is possible between two different strands of bacteria that were mixed together. So how does transformation occur like that naturally? Some bacteria have surface proteins that bind to DNA in the environment and transport it into the cell. And then inside the cell, the new DNA sequence is compared to the bacterium's DNA. And if it's similar enough, it can be recombined with the bacterium's DNA. If it's not similar, the new DNA gets broken down. So in research, scientists are able to use transformation to study pieces of DNA by replicating it safely and quickly in a non-pathogenic host bacteria. So for example, the human genes for insulin have been transferred into bacteria to produce insulin protein. So how do they do that? I just explained that if DNA is too different, then it gets broken down. So how do scientists transform bacteria to study DNA that's very different from the bacterium's DNA? The answer is that scientists transform bacterial cells with plasmid DNA. So a plasmid is a small extra chromosomal, meaning that it's outside the bacterium's chromosome. It's an extra chromosomal DNA molecule within a bacterial cell. So in this picture, the bacterial chromosome is represented by the red squiggly lines, and the plasmids are outside the bacterial chromosome. They're separate, small, circular DNA molecules inside the bacterial cell. They're small, circular, double-stranded DNA molecules. They're physically separated from the bacterium's chromosomal DNA, and they can also copy themselves independently of the bacterial chromosome. Plasmids are not essential for bacteria to survive. Bacteria don't necessarily have to have plasmids. They could possibly just have their chromosomal DNA and that's it. So they don't have to have plasmids. But the plasmids sometimes confer advantageous traits that help them to survive. So for example, the plasmids could provide advantages for the bacterial survival, like antibiotic resistance. So you can see in this picture, the plasmid has resistance genes for certain antibiotics, and that gives the bacteria an advantage of being able to survive in the presence of those antibiotics. So it's an advantage to the bacteria being able to survive and reproduce. And those plasmids are an ideal way of introducing different DNA into the bacteria. And there are several reasons why they're ideal for that purpose. First of all, they're small, so they can enter small pores in the bacterial cell membrane. Secondly, they have an origin of DNA replication, and that's the sequence of bases at which the DNA replication begins. And because they have that origin of replication, the plasmids can copy themselves, and they can do so independently of the bacterium's chromosome. The third reason is that it's easy to introduce new DNA, or the DNA that you want to study, into plasmids. In order to introduce that DNA that you want to study into a plasmid, the plasmid is first cut open with a restriction enzyme, or an endonuclease. And in this picture, that's shown as the scissors in the picture below. So the restriction enzyme recognizes a specific sequence of the DNA in the plasmid. And that specific sequence is called the endonuclease recognition site. And it cuts the plasmid open at that specific site. And then the new DNA fragment is inserted. So another enzyme called DNA ligase is used to join the ends of the DNA fragment, which is represented in green there, to the ends of the plasmid, so that that circle of the plasmid is recreated. But now you have a plasmid with a piece of DNA that you wanted to study. So now when the plasmid copies itself, it makes copies of that desired DNA too. So this is called cloning, producing identical copies of the original. In this context, in the context of DNA molecules, cloning means putting DNA into a living cell in such a way that the DNA will be maintained and copied by the cell. So talking about that example of the gene for human insulin again, being cloned into bacterial cells. And when that's done, the cells can then produce large quantities of human insulin. So the insulin gene is taken from a human cell. It's cut with the restriction enzymes. The plasmid is also cut with the restriction enzymes, and then that gene is inserted into the plasmid. The plasmid can then be transformed into the bacteria and grown in culture to produce large quantities of the insulin protein. And there's one more reason that plasmids are an ideal way of introducing different DNA into bacteria, and that is that plasmids can have selectable markers. So when we do a transformation experiment and try to put a plasmid with the DNA that we want to study inside a bacteria, only a small fraction of those bacteria in the experiment actually take in the plasmid. So how do we know if the bacteria actually received the plasmid? So you're going to have some bacteria that have just their normal chromosomal DNA, but some that have the plasmid that we tried to insert that actually did go inside the bacterial cell. So how do we know which ones are which? And then, how do we know if that plasmid actually contains our desired DNA fragment? How do we know that when we cut the DNA fragment and insert it into the plasmid, that it actually worked? The answer is that scientists insert the desired DNA into a plasmid that contains selectable markers. And then they use antibiotics and other chemicals in the media, in the plates, to select for them. So there's two types of selectable markers that we use in our lab. The first is a plasmid that contains a gene for antibiotic resistance. So, for example, the gene for beta-lactamase encodes an enzyme that cleaves certain antibiotics, and that makes them resistant to that antibiotic. So a bacterium that received the plasmid with that gene would be able to grow in the presence of the antibiotic. And the other cells that didn't receive the plasmid would die in the presence of the antibiotic. So if you're growing your cells on a plate that contains antibiotics, you're selecting for the cells that have been transformed. So now you know that all of your growing bacterial cells do have the plasmid. So again, using antibiotics as selectable markers, the host cell DNA naturally doesn't have the resistance gene, but the plasmid that you're trying to insert into it does have the resistance gene. So you try to put your plasmid into the bacteria, and you use antibiotics to select for those that actually took it in. But our second question was, how do we know if the plasmid has our desired DNA actually inserted into the plasmid? And the way that is done is by using a plasmid that contains a color marker gene. So for example, the gene for beta-galactosidase encodes a protein that cleaves the sugar lactose, but it will also cleave a similar link. So researchers developed a chemical called XGAL that turns dark blue when it's cleaved by the enzyme beta-galactosidase. So if you grow bacteria containing a plasmid with the gene for beta-galactosidase on a plate with the chemical XGAL, you would get blue colonies. Scientists then developed a plasmid where the desired DNA gets inserted within the beta-galactosidase gene, and then it just disrupts that gene so it no longer produces the blue color, and it gives you white colonies. So if your plates have ampicillin, which is an antibiotic, and XGAL in the media, you would get both blue and white colonies on the plate. The blue colonies would show you that that beta-galactosidase gene is still intact and your desired DNA fragment did not get inserted there. But the white colonies would show you that your desired DNA was inserted in the beta-galactosidase gene and disrupted it, which made it unable to produce the blue color anymore. So to summarize selectable markers, the antibiotic resistance gene in the plasmid assures you that all of the colonies on your plate contain the plasmid, because if they didn't, they wouldn't be able to survive the antibiotic. And then the color marker gene in the plasmid assures you that the white colonies contain the desired DNA, because it was inserted into the plasmid and disrupted the gene that's responsible for the blue color. So in the end, your white colonies growing on an ampicillin and XGAL plate are your successfully transformed colonies. Now let's talk a little bit about how that plasmid actually gets inside the bacterial cell. First of all, bacterial cells need to be made competent, meaning in a state in which they're able to take up the DNA or take up the plasmid. So a DNA molecule and the cell membrane are both negatively charged, so under normal conditions they would repel each other. But if the cells are soaked in calcium chloride, which is positively charged, it makes it possible for them to come close together. After the cells are soaked in calcium chloride, the process to get the plasmid inside the bacterial cell membrane is as follows. First, the plasmid and the cells are put together in a tube and placed on ice to allow the plasmid to come in close contact with the cells. Then there's a heat shock of the cells that causes the pores in the cell membrane to widen and allows that plasmid to enter the cell membrane. The cells are then returned to ice to close the pores and retain the plasmid inside the bacteria. And then nutrient-rich media is added to the cells. So that gives the cells a chance to recover. It allows them to start reproducing, and as they do that, they start expressing the DNA from the new plasmid. So the cells need to express that DNA because it has the antibiotic resistance. So in order to survive on the antibiotic plate, it needs a chance to start growing and expressing that DNA. And then finally, the cells are plated or spread out on the agar plate containing the ampicillin, which is the antibiotic, and the excal, the color for the blue-white selection. It's spread out on that plate to grow the transformed colonies.